Тепер за порядком денним є доповідь ДУАНЬ ЧЕНЬ, Тронгейм, Норвегія. Також у запису чи це в зумі? Запис. Сподіваюся, що ДУАНЬ ЧЕНЬ не піде таким шляхом, як пішов професор Курпіш.
Hi, my name is Duan Chen, a professor of experimental surgery at Department of Clinical and Molecular Medicine, Norwegian University of Science and Technology. So NTNU is abbreviation in Norwegian. The university is located at Trondheim. So the picture before the university main building, it is shows occasion of a PhD dissertation in which Professor Oksana Dajakiewicza from Lviv Medical University acted as faculty opponent on November 1st this year. And she invited me to give a talk at this medical forum, which is a great honor and pleasure for me. So the title of my talk is Translational Research on Gastric Cancer, Biomarkers and Drug Repurposing. As you may know, the methodology of translational research includes three components. The circle on the left shows bring the patients in the center of what we do, meaning what are the patient's needs for translational research. The circle in the middle shows bedside to bench and bench to the bedside, meaning the continued interaction and enrichment between research and clinical development activities. The circle on the right shows the clinical studies to impact research activities. So the goal of translational research is innovation from research studies to clinic development activities in humans. So this figure shows the survival of easy to treat cancer, the ETTC, and hard to treat cancer, HTTC, in Norway. So today I would like to present some of our results on stomach cancer, which is one of the HTTC in Norway. The photo on the right was the Atlantic route, and I like it because it is, to me, shows also there is always a hope. So this figure illustrating the gastric tumorigenesis involving carcinogens and risk factors and pathogenesis. I will not talk about this in details, but would like to highlight the transition from intestinal metaplasia, as shown by endoscopy and histology, to adenocarcinoma by CT and histology. And this was presented by Dr. Anderson, showing this picture in the occasion of his PhD dissertation. So we are currently looking for the potential biomarkers of the transition from gastric intestinal metaplasia to gastric cancer by transcriptomics in patient samples. So this figure shows the activities of 110 signaling pathways under the gene compared with gastric cancer. And we still try actually to find out which are the potential markers for the transition and why those are the downregulated pathways in gastric cancer. And we have built the gene to get gastric cancer biomarker networks that consist of 23 hub proteins. So this is a graphic summary of this study so far on potential biomarkers of gastric intestinal metaplasia to adenocarcinoma. Both surgical and endoscopy biopsies were collected for gene expression analysis. The preliminary results shows biomarker network and tissue resident stem cells and chromosome instabilities. And the biomarkers network and tissue resistance were related to the Wnt signaling pathways. So we hope these studies will make contributions in identification of biomarkers for surveillance of gene and drug targeting for stopping the transition in the future. So this article was published 30 years ago, and we found it very interesting, particularly the table 2 shows gastric cancer 18 years after vagotomy was 0 in patients with gastric ulcer. So we wanted to test our hypothesis that vagotomy suppresses gastric tumor genesis. So accordingly, we made experimental design, including transgenic mouse model of spontaneously gastric cancer called INSGAS mice, a chemically induced gastric cancer in mice, and H. pylori infected mouse model of gastric cancer with vagotomy versus the sham operations at the different ages. The vagotomy was performed, included either bilateral vagotomy or unilateral vagotomy, which were performed by Dr. Zhou and Anderson. These are some results of the vagotomy in mice. Let's see, the tumor incidence in mice was dramatically reduced after vagotomy, and only on the denervation site, as shown in the mice with UVT. Stem cell markers include beta-catenin, CD44, DCRK1, Wnt signaling pathways were reduced after vagotomy. Next, by using the co-culture of stomach organoids with neurons, the muscarinic acetylcholine receptor 3 knockout mice, immunohistochemistry, and transcriptomics, we have found out the possible signaling pathways in gastric tumor genesis, like the following: acetylcholine acts on M3 receptors in the gastric stem cells to stimulate the YAP, TAS, Wnt, HIP signaling pathways. Furthermore, we performed metabolomics in gastric cancer mice with or without vagotomy in comparison with wild-type mice and analyzed the key metabolites in connection with metabolism. So we found that neuronal signaling moderate the metabolism of gastric cancer, as shown here. Vagotomy induces metabolic reprogramming in healthy gastric tissue and gastric cancer tissue. Glucose was metabolized through oxidative phosphorylations, OXPHOS, and glycolysis and TCA, produce ATP and lactate in normal tissues. Glutaminolysis, where Wnt, c-MYC pathways and TCA cycle take place in the gastric cancer. So vagotomy reduces lactate regeneration in normal tissue and induces a normalization of ATP production in cancer cells. So that was done by Hannalina Robinson. So with computational prediction of drug repurposing, we have found that the following the potential drug target with drug repurposing in both mice and humans. For instance, SNAP25 for botulinum neurotoxin A, for example, Botox. So accordingly, we treated the mice and the patients with Botox. As you can see, Botox was injected into the site. The tumor size and the pathophysiology parameters were reduced in mice, and tumor size was stabilized in patients for the duration of clinic follow-up. We have also performed the similar studies on other drug repurposing, such RED 001 and CPI 616 and ivermectin and PEITC. So in conclusion, our studies have shown the possible causal association between innovation and tumorigenesis of gastric cancer via Wnt signaling pathways and metabolic reprogramming within the tumor microenvironment, and the potential treatment of gastric cancer with the vagotomy and botulinum neurotoxin A. In addition, our studies have shown the methodology with focus on patients' needs and wet and dry lab research and clinical studies. The method of translational research, such as phenotypic screening, cancer discovery, preclinical screening, drug discovery, bioinformatics, proteomics, transcriptomics, metabolomics, cancer patients, and clinical trials, and for future precision medicine. I would like to take this opportunity to thank my colleagues and collaborators as shown in this list. Dr. Trimizo, Yusuke Kodama, Hannalina Robin, Yara Anderson, and Timothy Wang, and Dama. To Yoko Hayakawa. Thank you.
Доповідь закінчена. Ми дякуємо за те, що вчасно вона закінчена. Ця доповідь також буде на Ютубі, чи вона вже є.